Section 1. Molecular Scissors. An Introduction to Restriction Endonucleases. In the world of molecular biology, restriction endonucleases are like tiny, precise scissors. These enzymes, found naturally in bacteria, have revolutionized the field of biotechnology. They allow scientists to cut DNA molecules at specific sites, opening up a world of possibilities for manipulating genetic material. Imagine being able to cut a DNA molecule, which carries genetic instructions, at a specific location. Restriction endonucleases make this possible. These enzymes recognize short, specific DNA sequences and make cuts at or near these recognition sites. This ability to cut DNA precisely is the foundation for many genetic engineering techniques. Restriction endonucleases have become indispensable tools in modern biology. Their discovery marked a turning point in our ability to study and manipulate genes. From gene cloning to gene therapy, these molecular scissors have paved the way for groundbreaking advancements in biotechnology and medicine. Section 2. A Fortunate Discovery. Unlocking the Secrets of Bacteria. The discovery of restriction endonucleases was a serendipitous event that transformed the landscape of molecular biology. In the 1960s, scientists studying bacteria observed a peculiar phenomenon. Some bacteria could resist infection by certain viruses, while others were susceptible. This resistance was linked to the presence of enzymes within the bacteria that could degrade the invading viral DNA. These enzymes, later named restriction endonucleases, were found to act as a defense mechanism for bacteria. They protect the bacteria from foreign DNA, such as that from viruses, by recognizing and cutting it at specific sequences. The bacteria's own DNA is protected from being cut by these enzymes through a process called methylation. This groundbreaking discovery earned Werner Arber, Hamilton Smith, and Daniel Nathans the Nobel Prize in Physiology or Medicine in 1978. Their work laid the foundation for the development of recombinant DNA technology, which allows scientists to cut and paste DNA from different sources, leading to countless applications in biotechnology. Section 3. Precision Cutting. How Restriction Endonucleases Target DNA. Restriction endonucleases are incredibly precise in their action. Each enzyme recognizes a specific DNA sequence, typically 4 to 8 base pairs long, called a recognition site. These recognition sites are palindromic, meaning they read the same backwards and forwards. For example, the restriction enzyme EcoRI, isolated from E. coli, recognizes the sequence GATC, which reads the same on the complementary DNA strand. When a restriction enzyme encounters its specific recognition sequence, it binds to the DNA molecule and makes a cut. Some restriction enzymes make straight cuts across the DNA double helix, producing blunt ends. Others, like EcoRI, make staggered cuts, leaving single-stranded overhangs called sticky ends. These sticky ends are particularly useful in genetic engineering. Since they have complementary single-stranded sequences, they can base pair with other DNA fragments cut with the same restriction enzyme. This allows scientists to join DNA fragments from different sources, creating recombinant DNA molecules. Section 4. Genetic Engineering Powerhouse. From Gene Cloning to Disease Therapy. The ability to cut and paste DNA using restriction endonucleases has revolutionized biotechnology and medicine. One of the most widely used applications is gene cloning, which allows scientists to create multiple copies of a specific gene. This process typically involves inserting the gene of interest into a vector, such as a plasmid, which can then be introduced into bacteria for replication. Restriction enzymes are also crucial for gene editing techniques, such as CRISPR-Cas9, which allows for precise modifications to DNA sequences. By introducing a Cas9 enzyme program to target a specific gene, scientists can disable or modify that gene with high accuracy. This technology has enormous potential for treating genetic diseases, developing new therapies, and even engineering crops with improved traits. The impact of restriction endonucleases on biotechnology and medicine cannot be overstated. These molecular scissors have paved the way for groundbreaking discoveries and applications, from gene therapy for genetic diseases to the development of genetically modified organisms with enhanced traits. As our understanding of these enzymes continues to grow, so too will their potential to address some of the world's most pressing challenges.